So welcome to the 2018 Parasite Awards, or as they're sometimes but rarely called, the PSB Awards for Rigorous Secondary Data Analysis. I won't go into a lot of detail on the history of these awards, but the goal of the awards is to recognize individuals who have taken someone else's data and, in the words of Ido Friedberg, done some really cool shit with it. So without further ado, the people who help us recognize these people. So the selection committee this year was myself, Lana Garmeyer, Melissa Handel, Marilyn Ritchie, and Eric Turner. So please, if you see these people, thank them for the time and effort they put into reading these applications. For those of you who applied or who know people who applied, we had so much fun reading your applications again this year. So please, if you did not win, please submit again because we always enjoy reading them. And there's always a good chance in future years that you will be selected. From putting the award together, we know that getting people to this meeting is expensive, and we have sponsors who make this possible. So three different journals or publishers came together to put together the prize money for the Parasite Awards as well as the travel support. So I want to thank GigaScience, Scientific Data, and Springer Nature for making this possible. And then for this year, actually, we were supposed to have our junior parasite on site, but he had a family emergency, so we actually have him in video form. So I'm about to show you a video from Uri Ben David. He was our junior parasite recipient, and he did a project using patient-derived xenografts that we thought was really cool. I would tell you more about it, but I know his video is about to give you details, so I'll leave it to him.
Hi, my name is Uri Ben David. I'm a postdoc at the Cancer Program of the Broad Institute in Cambridge, Massachusetts. I'm really sorry that I can't be there with you today to receive the award in person. If not for any other reason than because it's currently 0 degrees Fahrenheit here in Boston, and being in Hawaii right now sounds like a really good idea. I'm honored and excited to receive the Parasite Award this year. I would like to thank Dr. Green and the prize committee for selecting me, and more importantly, for establishing this awesome award to celebrate the rigorous secondary analysis of data. Hands down, this is the coolest award I've ever received. In 2018, it's quite surprising that we still need to encourage people to reanalyze each other's data and to view this reanalysis of data as something positive and even essential in science. The work for which I'm receiving the award today reveals distinct tumor evolution trajectories in human patients and in mouse models. Patient-derived xenografts, namely the direct transplantation of human tumors into mice, have recently become a very common model system in cancer research. These patient-derived xenografts, or PDXs in short, are presumed to retain the genomics of primary tumors better than cancer cell lines. However, the immune-deficient mouse microenvironment is very different from the microenvironment to which tumors are exposed in the human patient. Therefore, a key question in the field is whether PDXs indeed retain the genomic features of the tumors from which they were derived. To address this question, one would need to study at large scale the evolution of human tumors upon their transplantation into mice. Prior to our study, no such studies existed, largely because the generation of PDXs is experimentally challenging, laborious, and expensive. Previous reports have focused on relatively small cohorts of PDXs and mostly concluded that PDXs were genomically stable. Our study challenges this conclusion and reveals that PDXs undergo extensive evolution during their initiation and propagation, that this evolution is different from the evolution of tumors in human patients, and that it can affect the response of PDXs to anti-cancer drugs. The paper, which was published in Nature Genetics in November 2017, was entirely based on publicly available datasets, and would have not been possible otherwise. We compiled a dataset of 1,100 PDX samples, about a tenfold larger than any PDX dataset reported before. Had we or anyone else tried to generate such a large dataset experimentally, this would have required multiple years of work and thousands of mice. Therefore, mining other people's data, aka being a parasite, was absolutely essential for carrying out this study. However, we couldn't just take advantage of previously published DNA sequencing data from PDX samples, simply because such data were scarce. Only about 200 samples were available. In contrast, gene expression profiles were available for almost 1,000 PDX samples from over 500 PDX models across cancer types. I therefore applied a computational approach that I had previously developed during my PhD to infer copy number changes from gene expression. By transforming these gene expression profiles into copy number landscapes, we suddenly had a treasure trove of genomic data that until then had gone untapped. Consequently, we were able to analyze the largest PDX dataset ever generated without performing a single experiment or sacrificing a single mouse. Our work shows that PDXs undergo rapid and extensive genomic evolution on their transplantation into recipient mice. This evolution is driven by clonal dynamics that often lead to the expansion of rare pre-existing tumor subclones. Genetic events that are common in primary tumors often tend to disappear throughout the propagation of PDXs, and this can affect the tumor response to specific targeted therapies. Our work thus challenges a common notion in the cancer research field, that PDXs are genomically stable. This work therefore has important implications for PDX-based modeling of human cancer. I think that this study represents the spirit of the Parasite Award, as it demonstrates the importance of looking at existing data with fresh eyes. We provide the entire dataset, including all genomic landscapes, so that other investigators can reproduce our study, extend the results, and even mine the data to ask additional and potentially unrelated questions. I look forward to seeing what other investigators may discover when they perform a tertiary analysis of these data. Thank you all once again, and I hope you enjoyed the conference.
So thank you, Uri, for applying. We enjoyed reading the paper, and just to let you know, we've sent him a prize package that includes, among other things, the cash prize, but also more importantly, these, which Brian will explain in more detail going ahead, but we've upped our game this year. Our senior Parasite Award winner this year for work in lateral gene transfer that we found really interesting and compelling is Julie Dunning-Hotup. Again, she has sent a nice video explaining her research. Since she was not able to make it, I guess her lab was just renovated and she's been moving into new space, so that's exciting. But it meant she couldn't be here for this. So I'll let her explain it in her prize video, and hopefully the audio will be a little louder for this one.
Hello, and thank you for your recognition of my group's work generating new hypotheses from existing data through secondary data analysis with the sustained Parasitism Award. I'm sorry I could not make it to Hawaii, where it is currently 73 degrees Fahrenheit, to accept the award, as instead I'm in the DC-Baltimore metropolitan area, where it is currently 23 degrees Fahrenheit. I wish I could be with you. Let me just tell you a little bit about the work that led to our receiving the award. My group focuses on lateral gene transfer, and more specifically, lateral gene transfer from bacteria to animals. One very famous example of lateral gene transfer is that of Spider-Man. Spider-Man was bitten by a radioactive spider, and that spider's DNA was transferred to Peter Parker, giving Peter Parker the spider's ability, the ability to shoot webs out of its wrists, as well as to climb tall buildings. Our work on lateral gene transfer really began with a secondary data analysis of the Drosophila ananaceae genome, and it culminated in this paper describing widespread lateral gene transfer from intracellular bacteria to multicellular eukaryotes, which was also recognized by Discover magazine in 2007 as one of the top 100 discoveries of the year. We were focused on Wolbachia, a parasitic and mutualistic endosymbiont of insects, other arthropods, and filarial nematodes. It has many hosts, some of which are shown here, including termites, leafcutter beetles, parasitoid wasps, dog heartworm, and the causative agent of lymphatic filariasis. Initially, I was collaborating with Steven Salzberg to examine Wolbachia genomes that had been fished out of Drosophila sequencing projects. In particular, I was interested in Drosophila ananaceae, where some of the contigs appeared to be from the Wolbachia endosymbiont, those on the second line of this figure, while others on the fourth line of this figure seemed to be chimeric fragments of both Drosophila and Wolbachia DNA. Initially, these were chalked up to be misassemblies or assembly artifacts. However, over time, we were able to validate the junctions between the Drosophila and the Wolbachia DNA, validating that either Drosophila DNA had moved into the Wolbachia genome or Wolbachia DNA had moved into the Drosophila genome. And through a series of experiments that I am not going to describe here, we were able to show that it was actually Wolbachia DNA that had moved into the Drosophila ananaceae genome. It was actually during the review process that one of the reviewers requested that we actually scan the trace repositories for evidence of further lateral gene transfers in the publicly available genome sequencing data from animals. At the time, there were a whopping 26 arthropod and filarial nematode genomes that have the potential of being infected from organisms infected with Wolbachia. 15 are organisms known to be infected with Wolbachia, and 10 of those organisms actually contain Wolbachia traces. Of those 10, 8 show evidence of having Wolbachia host lateral gene transfer. That suggests that 31% of potentially infected organisms have lateral gene transfer, and 80% of genomes with Wolbachia reads have lateral gene transfer. There are some caveats, of course. This is not a random sampling of organisms. Deposited traces at the time, and possibly even today, have been cleansed of bacterial traces, and not all genomes are deposited. However, it seemed like this phenomenon was much more widespread than initially appreciated. Today we understand that bacterial DNA can move into some animal genomes, bacterial DNA from Wolbachia as well as other bacteria. With that knowledge, we set forth to examine if bacterial DNA could move into the human somatic genome. Think of the genome of cells lining your intestinal tract that can be exposed to bacteria and their influences. To do this, we developed a pipeline where we can look for pairs of reads that are discordant in their taxonomy. In this case, where one read matches a bacterial genome and the other read matches a human genome. We set out to calibrate this pipeline using data, publicly available data, from HeLa, which is known to have an HPV integration in its 8th chromosome. We were able to reconstruct that viral integration into the 8th chromosome of HeLa using our pipeline. We then applied this pipeline to data, publicly available data, from the TCGA project. This is the Cancer Genome Atlas. And in particular, we tested 9 different cancer types. And we were able to find evidence of bacterial DNA integration into acute myeloid leukemia samples and stomach adenocarcinoma. I'm only going to show you data from the latter. In stomach adenocarcinoma, we could find integrations of DNA from the bacterial 6S and 23S ribosomal RNA in the 5' UTRs of 5 genes found on 4 separate chromosomes. This was our first evidence that bacterial DNA may integrate into the stomach cancer genome. We were not able to validate these results because we could not get access to the samples that were used to generate this data. However, we were able to parlay it into further funding in order to generate our own data sets to try to validate these findings. And that work is ongoing. We were also able to use the same pipeline to pull out reads that are putatively from the microbiome of these tumor samples. Those are the red reads, where both reads mapped to a bacterial genome. In this case, there were numerous bacteria that were associated with multiple samples that are believed to be contamination. In particular, Propionibacteria, Staphylococcus, and Mycobacterium. However, there were other organisms that could not be attributed to obvious contamination of samples, and those are potentially microbiome associations with cancer that can be examined further. Our work on bacterial DNA integrations into the human somatic genome has been funded through an NIH Director's New Innovator Award, as well as an NIH Director's Transformative Research Award. The work on Drosophila ananaceae has been funded by the National Science Foundation, which continues to fund us to develop tools that enable others to detect lateral gene transfer and publicly available genome sequencing data. Our work has also been generously supported by many of the other organizations shown here. I am very thankful for the individuals in the left column who have conducted much of the research I mentioned here, as well as my many collaborators, including those listed here. I'm also very grateful for having a very supportive family who are shown here, my husband and my 2 sons. Thank you.
So I want to take the opportunity now to introduce Brian Bird, who's a physician scientist from Michigan, who's leading up an effort that we called the Symbiont Awards, and I'll let him take it away and explain what he's been up to.
Thanks, Casey. So I wrote down a few thoughts I'd like to share to frame what the Research Symbiont Awards are. So first, I'd like to thank our sponsors, Scientific Data and Springer Nature, who helped to make the award and this event possible. So the Research Symbiont Awards is a new concept, and I don't know about you, but I like to understand the context in which new ideas were developed. So to that end, I'm going to try to give you a sense of why the Research Symbiont Awards exist. I've worked on clinical trials for about a decade, and I also use data that have been generated by other people in secondary analyses. And more recently, I've been involved with, and to some extent have led, some projects involving public data or publicly available data that we've been analyzing. And that work allows us to continue to make interesting new observations even while smart, hardworking people in my laboratory are developing novel precision medicine assays to be evaluated in clinical trials. So when an editorial was published that spoke of research parasites, I read it the same day it was published, and I realized that this was not the correct framing. It's not the correct idea. I'm active on Twitter, where I found Casey Green and other creative data wizards talking about this idea of research parasites in very strong and very funny terms. And I noted the origin of the Research Parasite Awards, which I thought were right on, and I got a laugh out of it. But I also saw a gap. Specifically, in order for there to be successful, thriving reusers of data, they have to be successful, thriving people who choose to generate data and then choose to share it with the research community. And although many people generate high-quality data, in some fields, including my own, only a small number choose to share it. So I was aware that some people working in various fields, including my own field, have felt threatened by demands for data sharing, a proposition that in their worst fears would stifle their careers and most of all diminish their ability to impact and change the world. And I'm embarrassed to say that part of how I know that is that I also felt the same trepidation about sharing data before I reached a different view several years ago through discussions with my brother. A computer scientist and a highly enlightened spirit, he changed my perspective through a combination of logic, appealing to my better self, and the time-worn tradition of older brothers making fun of me and my untenable view. So although I now know that sharing data amplifies and enlarges one's impact on the world and in no way diminishes one's impact, I also recognize the trepidation of peers as familiar and very human. Notwithstanding all the reasons it's right to share data, it takes courage to be a first mover in a field in which successes have been achieved under the operating rule, it's mine. So on Twitter, I proposed that beyond promoting and rewarding data reuse, it's important to promote and reward data generation and sharing. Data generation, sharing, and optimal use are inseparable. They inform each other. So I tweeted something to that effect to a muted and somewhat skeptical response, but with some support from what I was saying from Casey and others, a few others. And soon enough, Casey contacted me and said, "Brian, would you like to work with me to start an award recognizing those who generate and share data?" And I said, "Yes, let's do it." I'm proud to have assembled a wonderful, diverse committee to help judge the nominations and select the winner of what we're calling the Research Symbiont Awards. And you can see the members of the committee here. I'm the chair. Amanda Haddock is part of the committee, Rebecca Riggins, Leslie Vachal, and Ethan Weiss. It's our intention to help foster an ecosystem in which generating data, sharing those data in highly reusable formats, and creative reuse of data are not competing with each other, they're complementing one another, as I believe they naturally do. At the level of an individual project, scientists can and will analyze the data they've generated without including any other scientists. And at the level of individual projects, scientists can and will analyze publicly available data without involving those who generated the data. These lone wolf activities highlight the fierce independence of the scientific mind, which is a crucial part of what makes the human mind so capable of changing the world. But at the level of an information ecosystem, optimal generation and use of data will ultimately happen when scientists also engage that other aspect that makes the human mind so capable, the social and affiliative part of who we are as humans that makes us concerned with making sure all important activities, ours and others, thrive. We hope that the Research Symbiont Awards help to foster great mutual respect between scientists who work in different ways. And I hope the awards cultivate a sincere desire on the part of scientists of all stripes to see each other succeed and to help each other succeed in changing the world. As Jacob Bernowski said, tolerance among scientists cannot be based on indifference, it must be based on respect. At the level of an information ecosystem, those who painstakingly generate data need people who work hard to analyze the data in new and creative ways. And data analysts need successful scientists who work hard to generate new data. The only rational way forward is to treat both of these styles of research as indispensable and to recognize that whatever approach or approaches we personally use, we rely on each other's continued success to ensure human progress. This is what we mean by symbiosis. There are two Research Symbiont Awards, one awarded to a more established scientist working in any field, called the General Symbiosis Award, and one awarded to a more junior scientist working specifically on a human health project, called the Early Career Clinical Research Award. These are the award criteria, and I would highlight that data sharing beyond what was typical in the field was considered essential. And in addition, the richness of the data and the extent to which it was annotated in a way that would invite reuse was considered to be essential. The winner of this year's General Symbiosis Award for a more established researcher is Dr. Morgan Ernst. Dr. Ernst was unable to join us, unfortunately, but she recorded a message for us. Before we see the message, I want to tell you a little bit about her. When she was a graduate student, she painstakingly extracted from thousands of papers a large data set describing the life history traits of over 1,400 mammal species. And she shared the data, a move that I think would have been contrary to the advice many people would have given her. The data are clearly valuable as they've been reused time and again. The data paper has been cited over 120 times, and the data have been used in papers published in Science, Nature, Proceedings, and National Academy of Sciences, among many other journals. She went on to collect other data sets and again released them for use at a time when this was not in the norm in her field. She was establishing a new norm. We'll hear from her in a few minutes by video. But first, let me introduce Dr. Fabio Zanini, who is here today. Dr. Zanini is the winner of the first annual Early Career Clinical Research Symbiont Award. He is an example for the next generation of researchers. Having assembled a large set of data regarding the evolution of HIV in a longitudinal patient cohort, he worked to code a system that would allow excellent sharing of the data. The website is absolutely impressive, and you should take a look at it if possible. And without further ado, I'd like to hear from Dr. Zanini.
Hi, everyone. I'm not used to getting prizes very much, so I'm a bit nervous. I would like to thank, first of all, Brian Bird and Casey Green and the Symbiont Selection Committee for their efforts to support the awesome cause of data sharing in biomedical research. And I agree in full with what Brian just said, that it's both hard, but it's also essential. And then I would like to thank the organizers of the PSB, obviously, for hosting the award ceremony at such a great venue like here in Hawaii, which is just fantastic. So 2 years ago, in my last year of grad school, I created a web application on the human immunodeficiency virus, also known as HIV. And as you may know, HIV is a chronic disease, and it's difficult to treat, in part because it can mutate in many, many different ways. My then advisor, Professor Richard Neher, who is now at the Biozentrum in Basel, Switzerland, and we had a collaboration which was fantastic with Professor Jan Albert and his group at the Karolinska Institute in Sweden. And together we collected almost 100 longitudinal blood samples from 11 HIV-infected people. And we sequenced the whole genome of HIV at high coverage. And by doing that, in around a year, I think, we soon had around 100 million sequencing reads and could track common and rare mutation in the virus across 15 years of infection. So there was so much information hidden in those sequences, and using that we were able to write a paper in eLife in 2015, and then there was more data, so we wrote a second paper, and then there was still more information, so we wrote a third paper and a fourth paper and so on. And now the funny thing is that none of these papers brings me here today. Instead, while scrambling and rushing to get the first manuscript in place, instead of focusing on typing the manuscript, I decided to put together a web page that would make the dataset available to other scientists in the most open way possible. And I'm deeply grateful to both Professor Neher, my advisor, and Professor Albert, our collaborator, for bearing with me during that time, since I was supposed to be writing instead. And it's thanks to their patience and support that I can be here today. And the rationale for wasting, quote unquote, around 6 months of my time on a website while manuscripts were waiting was the following. I thought, well, even if my data analysis is not so good, which most likely it's not, there's lots of smart folks out there that can do it better than me. And this way, maybe the HIV patient's efforts and pain is not wasted, no matter how good or bad I am. And I think this is the reason why I'm so grateful to Brian and Casey for pushing the scientific community and all of us towards a more widespread data sharing, because modern biomedical research is really like Lego bricks. Each brick is quite useful, but really a whole lot more can come out if we are able to put them together in a standard and open fashion. And thank you very much for your attention.
So last year, the winner of the Parasite Awards won a lamprey. And in honor of the Research Symbiont Awards, we've upgraded the prize to include a trout, and it is magnetic. If you need a leather lamprey, there's someone in Latvia who will make them for you. Casey can tell you who.
Thank you so much. It's awesome what you're doing there.
Congratulations.
Thank you.
Well deserved. Thanks. Well, and before we show Dr. Ernst's video, I simply wanted to mention that the nominees were all wonderful, and it was a very hard decision. So we're grateful to everybody who applied for the award. So let's take a listen to what Dr. Ernst has to say.
Hi, I am very sad that I could not be with you in person to accept the first Data Symbiont Award. My daughter, who made an eloquent argument for why she needed to escort me to Hawaii to help me accept my award, is also very disappointed that I could not make it. While I am sad I can't be there, I am deeply honored to be receiving this award, and I am already enjoying the official Data Symbiont trophy. More than anything, however, I am deeply excited that this award exists. Data is one of the most valuable commodities we have in science. As a result, many scientists often feel that it is something to be carefully guarded by the group collecting it. But this value also means that science benefits most when data is openly shared. And we know that science can do amazing things with open data. GenBank has transformed the way we think about and analyze genetic data. NASA's satellite images have transformed and driven research in a variety of fields, from archaeology to climate science. And individually collected data sets have transformed fields like machine learning and statistics by becoming the canonical test cases for developing and comparing new methods. But all of this is only possible when people make their data openly available. Creating a culture of data sharing is a key component for motivating people to share their data. Creating this culture will involve a number of changes in how we conduct and credit scientific research, including all of its products, from the raw data to the software that analyzes it, to the ideas and understanding presented in papers. I think that awards like this one are an important step in creating that open data culture by signaling that data sharing is important and valued by the scientific community. So, thank you for the honor. Thank you for making this award. And most importantly, thanks for all the fish.
So if you see Dr. Ernst and when you see Dr. Zanini, please tell them congratulations. I'd like to thank, again, the sponsors, PSB, the committee that selected the winners, the audience, and above all, the people, including all of the impressive nominees who are working hard to change and improve the culture of science.